Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Keratinization. From this word, what we can derive? It is a process of formation of keratin. That's why it is called keratinization. It is also known as cornification. The word cornify, it is indicating a corneocyte, a cell in stratum corneum. That is formation of a stratum corneum, also known as cornification. Now we will see what is the definition of keratinization or cornification. It is a process of keratinocyte differentiation. That is, a corneocyte will change its size, shape and composition while forming into various suprabasal layers. A keratinocyte takes birth in the basal layer and it will form various suprabasal layers, finally resulting in the formation of keratin-rich permeability barrier, that is stratum corneum. The justification of these two words came here. Keratin-rich layer is getting formed, keratinization. Ultimately, stratum corneum is getting formed, cornification. It includes molecular events. What is meant by molecular events? Events which are occurring at the molecular level, that is inside a cell, such as formation of keratin and stocking up of keratin intermediate filaments mediated by filaggrin. Filaggrin is filament aggregating protein. It is one of the component of keratohyalin granules. Along with it, in keratohyalin granules, lamellar, involucrin, like that, many proteins are there. Next, formation of cornified cell envelope in stratum corneum. Clear? Formation and stocking of keratin intermediate filaments, formation of cornified cell envelope. We will see one by one. First, we will see what is keratin. Keratins are intermediate filaments. What are these intermediate filaments? Just like bones, which form skeleton of a human body, mammalian cell also have skeleton. Skeleton of a cell is called cytoskeleton. They may be either actin containing microfilaments, tubulin containing microtubules, or either intermediate filaments. This keratins comes under intermediate filaments group, which is present in keratinocyte, forming cytoskeleton within the cytoplasm. Clear? Keratins are intermediate filaments. They are nothing but cytoskeleton of keratinocyte. There are total 54 number of human keratins classified based on pKi, that is based on pH, into type 1 acidic, type 2 basic, and based on amino acid composition and consistency. They are divided into soft keratin or epithelial keratin found in skin, hard keratin or trichocyte keratin found in hairs and nail. Soft keratins are rich in glycine and serine. Hard keratins are rich in cysteine and proline. Cysteine is very rich in sulfur and it also contains disulfide bonds. This is the reason for its hard nature. You can clearly see how hard a nail is and how resilient a hair is. This is because this mechanical resilience is because of rich sulfur and disulfide bonds in cysteine, which is provided by hard keratin. In contrast, our skin is very soft because it contains soft keratin, which is having very low content of cysteine. That means indirectly it is having very low sulfur and low disulfide bonds. That's why our skin is soft and hairs are very hard. Coming to structure of keratin molecule. These keratins share a common domain structure along with all other intermediate filaments. What is that domain structure? It is nothing but alpha helical rod domain. To remember, to easily study this keratin molecule, just remember A, B and C. A is first alphabet, that is head region of the protein. A for amino terminus and B is for body. Body is made up of alpha helical rod domain, which is made up of 310 amino acids. Keratin is a protein. Proteins are nothing but polymers of amino acids. Here you can see 310 amino acids are there in alpha helical rod domain. And finally the alphabet C. At the end, tail. C for carboxy terminus, tail region of the keratin. This head region is flanked to the alpha helical rod domain by initiation motif. Same way, tail is flanked to the alpha helical domain by termination motif. Clear? A, B, C. A for amino terminus head, B for body alpha helical rod domain, C for carboxy terminus tail. And head is attached by initiation motif, tail is attached by termination motif. Finally, you can see here 4 orange boxes, 1A, 1B, 2A, 2B. These are 4 helical segments which contain repeats of 7 amino acids. That's why it is called heptad repeats. Hepta means 7. These amino acids participate in hydrophobic interactions with another keratin. These proteins will hate water. That's why they are called hydrophobic. Phobia means fear of water. These 4 helical segments are connected to each other by 3 non-helical linker segments, that are L1, L2 and L3. Here you can see linked by L1, L2, L3, 3 non-helical linker segments. Clear? Finished. Keratin structure is finished. 4 helical segments linked by 3 non-helical linker segments. L for linker, L1, L2, L3. These keratins forms obligate heterodimers. Obligate means they form only heterodimers. One keratin monomer of acidic type interlinks with another keratin monomer of basic type. 2 monomers linking together with heptad repeats hydrophobic bonds, forming dimer. 2 monomers together called dimer. Each is different type. One is acidic, one is basic. So they are called heterodimers. Many such heterodimers will link together to form tetramer. These tetramers will link with many such another tetramers to form a filamentous structure, ultimately forming cytoskeleton of a keratinocyte. They won't stop here. They will also anchor to desmosomes at one end and to the hemidesmosomes at the other end, forming a scaffold of mechanical proteinaceous structure which is connecting all the epidermis and dermis together tightly. Here you can see clearly keratin intermediate filaments, also known as tonofilaments. At one end they are anchoring to desmosomes. Desmosomes connect all the keratinocytes. Then at another end they are anchoring to hemidesmosomes, which connects basal layer to dermoepidermal junction. Indirectly, desmosomes, keratin intermediate filaments, hemidesmosomes, all 3 together forms a scaffold of connecting network. Hence, keratins will form filaments by forming heterodimers, tetramers and then functional intermediate filaments, which forms cytoskeleton ultimately. Now, anatomy of keratin finished. Now we will see applied aspects of keratins. Keratins have varied expressions according to the location. Coming to epidermis, in basal cells, keratin 514 heterodimer will be present. I already told you they will form only heterodimers. Here you can see keratin 5,4. If these keratins are defective, it will lead to genodermatosis. Here keratin 514 defect will lead to epidermolysis bullosa simplex. S for simplex, S for superficial blistering disease. Either it may be localized, generalized intermediate, generalized severe. If only keratin 5 is defective, it will lead to EBS with mottled pigmentation or with migratory succinate. Mottled means just like paint splashed on a wall, it will be patchy, irregular pigmentation. If keratin 14 is defective, it will lead to epidermolysis bullosa simplex, autosomal recessive form. Mostly this genodermatosis because of defects of keratins. Keratinopathies are autosomal dominant. Here only keratin 14 thing is autosomal recessive. Genodermatosis with reticulate pigmentation. Reticulate means fishnet-like pattern. Keratin 14 defect leads to dermatopathia pigmentosa reticularis. Keratin 5 defect leads to Dowling-Biggos disease. Finished. Remember, basal layer, epidermolysis bullosa simplex. Then suprabasal layer, it leads to ichthyosis, keratinopathic ichthyosis. Only basal layer contains keratin 514. Rest of all the layers contain keratin 110. During the process of differentiation, this keratin shift will occur. Keratin 110 defect leads to epidermolytic ichthyosis. Remember, it is main. Next, it will also lead to annular epidermolytic ichthyosis, epidermolytic nevi also. Keratin 1 defect leads to ichthyosis with Macklin type of keratinopathic ichthyosis. K2. In all suprabasal layers, keratin found is K110, but in granular layer, keratin 2 also will be present. If it's defective, it will lead to superficial epidermolytic ichthyosis. Keratin 10 defect leads to autosomal recessive epidermolytic ichthyosis. Same in keratin 1, in keratin 5, 14 defect leads to autosomal recessive form. Here also in keratin 1, 10 leads to autosomal recessive form. Rest of all are dominant. In palms and soles, it will lead to palmoplantar keratoderma. Name itself suggesting palms and soles. Palmoplantar keratoderma. In primary ridges, keratin 1,9 will be present. If it's defective, it will lead to epidermolytic palmoplantar keratoderma. If keratin 1 is defective, it will lead to non-epidermolytic PPK. Secondary ridges contain keratin 6A16, keratin 6B17, which leads to pachyonychia congenita. If keratin 17 is defective, it will lead to steatocystoma multiplex. In hair, so many keratins are there. Here I have mentioned only thing which gets defective, which leads to monolatrix. In this, only the star marked one, if you remember, it's more than enough for applied aspects of keratinopathies. Coming to epidermal kinetics. The thickness of the epidermis, number and size of the keratinocytes will remain constant. This constancy is maintained because loss of cells is equal to the production of cells. If a cell is lost, immediately another cell will replace that cell, hence maintaining a constant kinetics. That is, thickness is same, number and size of the keratinocytes will be same. From where these cells are produced, new keratinocytes are produced. That is epidermopoiesis, where it occurs. It occurs via keratinocyte stem cells in basal layer, which are present in clusters, mainly in the bulge region of hair follicle, which will give rise to all the rest of all the layers of epidermis. These proliferative cells will enter cell cycle with 4 phases. Just study cell cycle in this diagram itself, it's more than enough. Don't study in IADVL. There it's very confucius and some mistakes are also there. For example, in IADVL it's given G1 phase is also interphase, but actually interphase is a phase between 2 mitosis. Here you can clearly see it includes G1, S and G2. Entire these 3 phases comes under interphase only. So no need to refer any book, just study this cell cycle here, which has 4 phases. Simple. One is mitosis phase, where division of nucleus, division of cytoplasm, and a parent cell will divide into 2 daughter cells. After mitosis, G1 phase. Because it is coming after mitosis, it is called Post-mitotic. G means growth phase. Post-mitotic growth phase. I have star marked here because maximum growth of the cell occurs in this phase where proteins will be synthesized, RNA will be synthesized. Assembly of RNA is nothing but DNA synthesis, which will occur in next phase, S phase. DNA synthesis will occur. And then cell will enter G2 phase. G means same growth. Before mitosis, this growth is happening. That's why it is called pre-mitotic growth phase, where no much of growth will occur, but some growth will occur, some protein synthesis will occur. Mainly what is the function of this G2 phase is, if any defective DNA has produced here, that DNA repair will happen here in G2 phase. Before dividing, DNA, any defects are there, repair will undergo here in G2 phase. After DNA repair, DNA synthesis, now the cell is ready for division, that is mitosis. After mitosis, we will get two daughter keratinocytes. These two daughter keratinocytes have three options. One, it can re-enter the cell cycle and continue post-mitotic growth phase, again S phase, G2 phase, and then mitosis, and then again produce new keratinocytes. Another option is it can enter quiescent phase, that is Q0 phase. It can take a break from this cycle, maintaining its potency to divide when appropriate stimulus comes from the environment. Until then, it can remain quietly without entering cell cycle and getting tired. Then last option, terminal differentiation. This is the favorite option for majority of the keratinocytes. They will not enter cell cycle again. They will not be quiet and wait in quiescent phase. They will decide to differentiate itself by losing all its connections from the basement membrane zone and go on to a tour of all the layers of the epidermis, ultimately reaching a dead layer called stratum corneum, and it will die ultimately. This process is called terminal differentiation. Just like many humans who pray for God for moksha because they don't want to rebirth again as a human and continue human life cycle, they want moksha. Same here also, it don't want to enter life cycle and produce another keratinocyte. It will decide to die either, breaking all the connections from the basement membrane zone. Stem cells are present in stratum base cell. So they are cells of stratum base cell. They will contain keratins 5 and 14. Here this is the cell cycle which produces new keratinocytes continuously. Why new cells have to produce continuously? Because majority of the cells are going on a tour to die to stratum corneum and they will shed off from the body. Thus that last cells have to get replaced by new cells. That production epidermopasis is taken care by this stem cells and via this cell cycle. Clear? Cell cycle, 4 phases, 3 options, again entering cell cycle, or quiescent phase, or our topic proper, keratinization, that is nothing but terminal differentiation. Who will regulate this epidermopasis? It is regulated by balance between stimulatory and inhibitory signals. Coming to stimulatory signals, these are nothing but growth factors like epidermal growth factor, transforming growth factor alpha, amphireglin. It is a major keratinocyte growth factor which is very specific to keratinocyte. All these 3 acts via epidermal growth factor receptor. Keratinocytes also produce some interleukins like 1, 6, granulocyte, monocyte, colony stimulating factor. They also stimulate epidermopasis. Reduced intracellular calcium also stimulates a cell to divide. In contrast, increased intracellular calcium or calcium levels will lead a cell to terminal differentiation. As calcium levels increase, cell will decide to differentiate. Vitamin D also same. Interferon alpha beta transforming growth factor beta. Beta is for inhibitory. Vitamin A normalizes abnormal keratinization. This beneficial effect of vitamin A is utilized by producing drugs called retinoids. We'll see in retinoids topic clearly. As I already told, majority of post-mitotic cells will decide to not to undergo any mitosis and become non-mitotic and lose integrin expression. Integrin is a transmembrane protein which attaches a basal cell to dermoepidermal junction. Now it has decided to lose its connection with basement membrane zone by losing integrin expression. Now it is free. It can go on a tour to all the layers of epidermis. Here you can see, thereby getting detached from basement membrane zone and move towards the skin surface to form stratum corneum. During this movement, they undergo a process of terminal differentiation in spinous layer. Now a basal layer cell, same cell have entered spinous layer. Now here they won't produce keratin 514 anymore. They will start producing keratin 110. We all must have heard this statement, be a Roman when you are in Rome. The same way, basal cell, now it is in spinous layer, so it will produce keratin 110, and because of increased calcium levels, it will also start producing woodland bodies in the upper layers. Now the same spinous layer cell has entered the granular layer. Here in granular layer, according to its requirement, here there will be so much of calcium levels, peaks of calcium will be there. It will force the cell to produce granules. In woodland bodies or in spinous layer, only woodland bodies are formed, only in the upper layer, but in granular layer, name itself suggesting us so many granules will be produced. Mainly two types. One is keratohyalin granules. Mainly these granules contain proteins which form cornified cell envelope, and lamellar granules. These are nothing but woodland bodies only. They will produce lipids mainly. L for lamellar granules, L for lipids, forming cornified lipid envelope. Where they will form these lipids? Exactly at the granular corneum interface. What is interface? Interface is nothing but junction. At the junction between granular layer and stratum corneum layer, these granular cells, woodland bodies, will come and fuse with plasma membrane, and then they will discharge their lipid content into intercellular spaces. Fuses with plasma membrane to discharge their lipid contents into intercellular spaces to form hydrophobic layer called cornified lipid envelope. Hydrophobic means it will repel water. It won't allow entry of water into our skin, forming an effective waterproof mantle. Mainly it contains acyl ceramides. That's why we will prescribe ceramide based moisturizers for atopic dermatitis patients. Here, if this lipids formation and cornified lipid envelope, if by chance getting defective because of ABCA12 transporter defect, it will lead to harlequin ichthyosis. Just remember, harlequin ichthyosis is related to lipids, cornified lipid envelope. From where lipids are being produced? From lamellar bodies. All these will be disrupted because if there is any damage to, if there is any defect in ABCA12 transporter protein. Now the granular layer cell released lipid contents. Now it is ready to enter stratum corneum layer, a dead layer. In the stratum corneum, these cells forms a highly insoluble cornified cell envelope. This envelope contains proteins cross-linked by transglutaminase 1. What are the proteins? Mainly keratohyalin granules. We have already seen keratohyalin granules are contributing to cornified cell envelope. We will see what are those granules. Filaggrin, mainly cysteine-rich protein. Involucrin, small proline-rich family containing cornifin, SPR1, and pancornulin. Plakins, mainly envoplakin, periplakin. Rest of all layers will have mainly desmoplakin, but here in corneocyte, envoplakin and periplakins will be there. And keratins, and remaining rest of the desmosomal proteins also. All these proteins are cross-linked by transglutaminase, forming a solid rock-like envelope, cornified cell envelope, which will be present deep to the plasma membrane. If in case there is defective transglutaminase 1, it leads to lamellar ichthyosis. Lamellar ichthyosis is less fatal and they can have a normal life. When compared to harlequin ichthyosis, where APCA12 is defective, it is very fatal. Ultimately, the final product of keratinization is a dead cell, which will be shed and replaced by new ones continuously. Everything is temporary, nothing is permanent. If this cell has shed, another new cell will replace its place. Here this dead cell is having cornified envelope packed with keratin intermediate filaments. This rich proteinaceous rock solid cell is compared to a brick. Corneocyte is compared to a brick and it is surrounded by a lipid envelope. Lipids are produced from woodland bodies at the junction of granular and corneum layer. Is compared to mortar. Both the brick and mortar together called as brick and mortar model of corneocyte, that is stratum corneum layer, forms an effective barrier. What a brick and mortar will do, they will attach to each other tightly, forming an effective barrier. This effective barrier formation will be finished by 24 weeks of gestation age. After this cornified layer formation, there is no need of periderm to protect from amniotic fluid. That's why periderm will shed off at that time. After 24 weeks, this keratinization will be continued throughout the life. Throughout the life, cells will be died, shed off and replaced by new ones. This brick and mortar, cornified cell envelope and cornified lipid envelope, also gets attached to each other by ester linkages, mainly by acyl ceramides. Epidermal turnover time for a normal skin is 52 to 75 days. What does this epidermal turnover time means? It means from birth of a keratinocyte to its entire journey through all the layers and then ultimately shedding off from the skin surface. This time taken is called epidermal turnover time. Whereas in psoriasis, entirely contrast. Within 3 to 7 days, it will finish. That means more and more number of cells are producing and pushing the old cells away. These old cells will not lose nucleus also. It won't have any time to lose nucleus also. With the nucleus only, it will enter the stratum corneum layer. That presence of nucleated cells in stratum corneum is called parakeratosis. Normally, this shedding of stratum corneum cell will not be visible to us. A number of cells will be naturally shedding off without any visibility. But in psoriasis, many cells will be produced within 3 to 7 days. So, that shedding of cells will be clearly visible. That visible exfoliation of stratum corneum is called scaling. What type of scales will be there in psoriasis? That is silvery white scales. Hence, we have completed the journey of a keratinocyte from its birth to its death. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. Das vidaniya.